Welcome to Club EvMed. Good to see people tuning in. Happy Tuesday at the almost the end of the semester for a lot of us. Apologies to those of you on the quarter system who still have another month. But you started later, so. Okay, let's give it a minute or two. Do we have other people still coming in, Mary? Okay, Mary Joy is our assistant at TRISEM who organizes all of these things, keeps everybody informed, keeps our website up to date. Thank you very much, Mary. We appreciate it. And good to see people tuning in. Kind of feels like things have slowed down. I'm just going to give a little bit of an introduction to the series, and then I'm going to turn things over to Vaughn Cooper, the editor-in-chief of the journal Evolution, Medicine, and Public Health, and he's going to introduce the speaker today. So my name is Charlie Nunn. I'm at Duke University. I'm in evolutionary anthropology and Duke Global Health, and I run the Triangle Center for Evolutionary Medicine. And it's great to welcome everybody to another episode, I guess, of Club EvMed. This is our Club EvMed conversations. We like to feature the latest work in the field, bring people together, learn something new, and also share perspectives. So we learn from one another, not just from the speaker. So thank you all very much for coming today. The Club EvMed series is organized, if I can get this to work, by a number of different centers, including TRISEM, but also a number of other centers around the world, and also by the International Society for Evolution Medicine and Public Health. I'm pasting all of this into the chat. Hopefully it's going there. I want to just highlight, of course, our journal, and Vaughn might say a few words about this as well, Evolution Medicine and Public Health, and also encourage everyone to come to our annual conference, the Society's annual conference in Kiel in Germany, and that's on July 28th to 31st. So anyhow, thank you guys for joining today. I'm going to turn things over now to Vaughn to introduce our speaker, Melissa.
Thanks, Charlie. Thanks for all the leadership and coordination you and your team have done on all of our behalfs and the EvMed centers. As Charlie said, I'm at the University of Pittsburgh in the School of Medicine, where we have our Center for Evolutionary Biology and Medicine. I just finished our EC meeting about a minute ago, and we're delighted to be a part of it. Continuing to recruit people in this area, which is exciting. And I'm always happy to point them to this journal that I now serve as EAIC for, the Evolution, Medicine, and Public Health Journal, which is the path by which I got to know our speakers' research, because Melissa Manis, who submitted a really thoughtful and engaging article that I enjoyed working with through the editorial process. I learned a lot, and I think it really is an example of showing how human practices can shape our microbiome and lead to short-term and potentially long-term effects. And this field is huge, growing, and always in need of the evolutionary biology perspective. So we're delighted that Melissa is contributing to that field by leading it and also by presenting for it. As for the EMPH journal, before I turn it over to our speaker, we have a call for articles that is roughly on this topic, and I'm going to post the direct link to that call here in the chat right now. And that's being made possible by a trio of guest editors. And I just want to point out to those of you who are watching now or maybe watching this recording, if there's an area that you see needs to be featured in our society journal, please don't hesitate to consider serving as an editor, proposing an issue. The load is relatively light, especially if you can share it among friends, and it really helps advance a particular area of evolutionary biology and medicine, again, in the support of our journal. So thanks. I will now turn it over to Professor Manis, who's joining us from the University of Texas at San Antonio. Take it away.
All right, thank you. Can everyone hear me okay and see my screen okay? All right. Okay, so hi everyone. Thanks for joining today. Looking forward to sharing some of my research with you, where we'll really be talking about this idea of microbial mismatch, and some of my work showing that common birth practices are associated with differences in newborn microbiome development. All right. So, I study the human microbiome, which we probably all know is this vast collection of microbes that are living in and on our body. These microbes are not just there hanging out, but they can really be considered as an important extension of our own human biology. Our microbiome is shaped by where we live, what we eat, who we interact with, and how we spend our free time. All of these factors then influence the types of microbes that we can acquire from the surrounding environment. And in turn, those microbes can influence our health by affecting our growth, immune function, and even our mood and behavior. And so, viewed through this lens, we can see that a lot of human biological variation is really the product of variation in the microbes that become intertwined with our physiology. And importantly, I think especially for this crowd, this variation can shape phenotypes that impact our health and survival, which makes the microbiome very relevant to considerations of human adaptation and evolution. Now, my research focuses specifically on the development of the microbiome in early life. And so, with this early life focus, my work is really articulating with the developmental origins of health and disease framework, which posits that human biology is quite sensitive to environmental inputs during this early life critical window. So, during this period, our biological development is very plastic or malleable in response to signals from the environment. And the microbiome is really coming online during this period and actually directing a lot of this developmental plasticity. So, as one example, we know that the development of the immune system actually relies on receiving instructions from the developing microbiome. So, experimental work has shown that if you raise juvenile animals in a sterile environment, so they're not exposed to microbes, they don't have their own microbiome, they also fail to develop a properly functioning immune system. What's really cool is some of these effects can be reversed, but only if those animals get exposed to microbes during that early life critical window and not after that window has closed. So, this, of course, is telling us that aspects of our biological development, including our microbiome development, are time sensitive. And so my research framework can really be summarized in this figure. I focus on the relationship between early life environments and health outcomes, and this potential mediating role that the microbiome may be playing along this pathway. More specifically, I ask, how do infants actually acquire and develop a microbiome, and how does this influence their health? So, as I think about this question today, an important thing to keep in mind is that as of right now, the scientific evidence tells us that human infants are pretty much born sterile, right? We don't think that there's major microbial exposure happening in utero. So, that means that the first opportunity for a major microbial exposure is actually during birth itself. So, we know that during vaginal delivery, newborns get exposed to maternal, vaginal, fecal, and skin microbes. Those microbes then seed the infant body and start to train the developing immune system. As some of you may know, there's been growing interest in this idea that C-section infants are lacking this typical microbial exposure, right? Since they're not interacting with maternal, vaginal, and fecal microbes in the same way during delivery. Since we've known for a while that C-section babies are a higher risk of inflammatory and autoimmune conditions, it follows that perhaps those health disparities are actually driven by initial differences in the newborn microbiome that is beginning at birth. And so previous studies have indeed found differences in important bacterial genera in the newborn gut, depending on birth mode, as well as various immune biomarkers that vary depending on birth mode, with a general trend of lower biomarker values in the C-section group. So some of these studies have really started to suggest this broader idea that birth mode can be creating initial differences in both the newborn microbiome and associated physiological development. There's another study that sort of went beyond just the microbes that were in baby's body, but also looked at what those microbes were doing. So one nice thing to measure is microbial metabolites. These are basically the bioactive compounds produced by our microbes that then circulate through our body, interact with our immune system, and give energy to our cells and tissues. So in this study, C-section infants were actually producing more butyrate, a particular type of microbial metabolite. They're producing more butyrate compared to their vaginally delivered counterparts. This was true at 3 months of age, even in models that adjusted for other practices and maternal health factors that may shape the microbiome. Why this matters is that butyrate has been linked to excess weight gain and obesity, so further suggesting the role of the microbiome in connecting C-section birth and later life health outcomes. Now, putting this into the evolutionary medicine language that many of us on this call are familiar with, these differences in the newborn microbiome, metabolites, and immune markers based on birth mode are really suggesting a type of mismatch, right? Where evolutionarily typical microbial seeding gets disrupted by C-section, a modern day medical practice, which may have negative impacts on infant development and health outcomes. Now, there are, of course, contexts and situations where C-section birth is unavoidable and is sometimes medically necessary, which has raised the secondary question of how can we mitigate this microbial mismatch that C-section may be producing. So in one study, researchers collected vaginal fluid on gauze and used this to inoculate C-section babies with maternal microbes immediately following birth. They then compared the developmental trajectory of the microbiome between the babies that were born vaginally, those born via C-section, and those who were born via C-section but received that experimental inoculation. And they found that overall, the C-section infants who were inoculated with maternal microbes, shown in these figures in green, actually had pretty similar microbiome developmental trajectories compared to the babies born vaginally, who were shown in blue. Over time and across different body sites, the C-section infants who were not inoculated with maternal microbes, shown in red, tended to display different microbial profiles. Another study did something similar, but now used human milk as a matrix for inoculating the C-section babies. So this actually involved a fecal microbiome transplant from mother to infant that was delivered through human milk. And this paper found that in the first few months of life, the abundances of multiple gut microbes were actually similar between the babies who were born vaginally and those C-section babies who received the fecal microbiome transplant. So those are the blue and black lines in these figures. And again, the C-section babies without the intervention in red tended to stand out. Now, at this point, you may be thinking that these studies are really just showing us differences in the microbiome early on. But what does this really tell us about health outcomes and about mismatch? I do think it's important to acknowledge that few studies are really designed to measure the microbiome and different health outcomes over time. There are a number of logistical challenges with sample collection, including the fact that the very nature of wanting to collect longitudinal health data means that it takes a long time. This idea of C-section as a mismatch and ways to mitigate it is relatively new, and so it just hasn't been tested extensively. But this, of course, makes it harder for us to understand the evolutionary importance of these early differences in the microbiome due to birth mode. One quick example of a study that addressed some of these shortcomings came out a few years ago, in which they recruited women who are having planned C-sections. They then used a blinded randomization process to give some of those C-section babies a gauze soaked in vaginal fluid, and then the placebo was just a gauze soaked in saline. The main outcome of interest here was a neurodevelopment score that was measured at both 3 and 6 months. And this study found higher neurodevelopment in infants who are inoculated with those maternal microbes at both 3 and 6 months. And they also found that the C-section infants who received that vaginal fluid inoculation displayed different gut microbiome trajectories compared to the control group. Those gut microbiome profiles were associated with the higher neurodevelopment scores. So, I think these studies are really interesting, but they do raise a very important question, which is why in the world is this happening? And how is infant biology so sensitive to these microbial exposures that begin right at birth? I believe that we can answer a number of these questions by taking an ecological and evolutionary perspective. So, community ecology tells us that phenomena like priority effects and community succession are really critical for shaping the dynamics of any type of community, including a microbial community. So, here we're talking about the first wave of colonizing microbes, really setting the stage for subsequent microbial community development. That's because the early arriving species can alter environmental conditions and resources in a way that's going to impact the later arriving species. So, continuing this example of C-section birth, it could be that the initial colonizing microbes are really setting the stage for a future trajectory of taxonomic and functional development in the infant microbiome, but that trajectory may be altered or decreased in the C-section babies compared to those who are born vaginally. In other words, the microbes that hit baby's body in the first few hours or days of life may really strongly impact future microbiome development. One example of this that's been explored more empirically comes from a study of Danish infants that focused on Bifidobacterium longum infantis, better known as B. infantis. B. infantis is an early arriving gut species that is transmitted through human milk feeding. B. infantis is a very cool microbe because it is well adapted to consuming some of the complex sugars that we find in human milk. So, milk is delivering both B. infantis and a food source for this species. So, this allows B. infantis to really blossom in the newborn gut and start to establish itself and train the immune system. Now, in the study, researchers found that B. infantis could outcompete other related species that were less proficient at breaking down those milk sugars. And when the authors transplanted the human microbes into a mouse model, they found that the order of arrival and the nutrient availability shaped microbial community development. So, without milk sugars present, other bifidobacterium species could become dominant in the community. But with the milk sugars present, B. infantis continued to dominate, even if it was introduced later. So this gives us a sense that there are really complex community dynamics that are at play when microbial communities are first being established in or on a host. So that was a lot of information about the infant gut microbiome, which has received a lot of attention in the literature. But our body is, of course, home to microbial communities at other body sites as well. A lot of my previous work has focused on the microbial communities of the skin and how that microbiome is developing in early life. So we know the skin microbiome plays an important role in immune activity, and it may also be connected to the development of the infant gut microbiome because human babies are so frequently putting their hands and their feet and the microbes on those body parts into their mouths. So, back in spring of 2021, I was recruiting newborns at a local hospital for part of my dissertation research. I was excluding C-section babies for all of the reasons that I just talked about, but I was also asking questions about antibiotics and general experience of giving birth in the hospital during the pandemic. And something that kept happening to me during this phase of participant recruitment was that mothers would agree to participate in the study, but they would tell me that I had to wait or come back to their hospital room because their newborn wasn't there at the time. And what ended up happening was that for a lot of these babies, they had actually already been taken out of the room to have their first bath. I became quite curious about this practice and started asking questions of the nurses and the physician who was overseeing my work at the hospital. And what I learned is that this newborn bath, which is quite common in many hospitals, was at this particular hospital something that just sort of happened to most newborns. I heard again and again that newborns were being bathed within the first day of life, but it was often earlier than that, just sort of based on nurses' schedules and a general perception and desire to get the bath over with so that mom could finally hold a clean baby. This struck me as really interesting, both from an anthropological and ecological perspective. Now, the skin is our largest organ, and we know that it's composed of different skin sites that provide different habitats for different microbes. This is part of why we see variation in the skin microbiome across body sites in adults. So, for example, your humid armpit is going to provide a different ecological niche for microbes compared to the dry skin on the back of your hands. We also know that hygiene practices impact the skin microbiome. We know there's an impact of hygiene products that we use on our armpits, and also an impact of washing our bodies with soap. As an aside, this is a finding that came from research back when I was a master's student in Charlie's lab in what feels like a lifetime ago. So, getting back to our newborns in Chicago, I had wondered if these newborn skin sites were actually more homogeneous because they were not yet as differentially sweaty, oily, or flaky compared to adult skin. So maybe we'd see more similar microbial communities across different skin sites. But I also wondered if the microbes that were trying to colonize the newborn skin would be sensitive to some of the environmental changes that were being induced because of this hospital bath. In other words, is the bath actually removing that first wave of initial colonizing microbes? And could this set the stage for a potential mismatch? So the results that I'm going to show you next are either directly from or are more of an extension of the paper that Vaughn referenced that was published in EMPH in 2024. This paper looked at the skin microbiome of the newborns that were recruited at this time point in the hospital and a subset of them who were followed up about 6 weeks later. We conducted 16S bacterial gene sequencing on these skin microbiome samples. So this characterizes the taxonomic composition of the microbes at the skin sites. And given what I was learning talking to families in the hospital, I included a question on my survey that just simply asked whether or not the newborn had been bathed prior to me swabbing them. All right, so as I mentioned, one of my initial questions was if we would see variation in microbiome profiles across the skin sites soon after birth, or if instead things would be more homogeneous because maybe early on skin is just skin. Interestingly, newborns who were sampled within hours of being born did display some variation in the diversity and composition of their skin microbiome. So we found that the armpit microbiome had the lowest bacterial diversity and had a different composition compared to the other skin sites. So the lower diversity of the armpit samples is shown in the box plots, and then below is a sort of cluster analysis that looks at the taxonomic composition of microbes in each of the samples. And you can see on the left at that top row, we're starting to get this cluster of these green armpit samples, which is telling us that there's similarity in the taxonomic composition of those samples. I'm not going to talk much about the paired infant samples today. I will just say that that body site differentiation strengthened throughout the first 6 weeks of infancy, which we might expect. So we see that cluster of green armpit samples is getting larger, right? More samples are clustering together. And we're also starting to see pockets of these orange samples, which correspond to the infant cheek. All right, so then wanted to understand which factors of the birth environment could be explaining some of this variation in the newborn skin microbiome, and if any of this was varying by skin site. So one of the things we did was use a redundancy analysis based on microbial relative abundances. And here we found that maternal antibiotics during delivery and infants having received a bath in the hospital before I swabbed them were statistically significant explanatory variables. So that is, they were explaining important variation in skin microbial communities. Now, one thing that I want to note is that on my survey, I asked mothers if they were aware of receiving any antibiotics during delivery. Some definitely said that they did. Others definitely said they did not, while others were all around unsure. I'm including this variable here for mothers who gave a definitive answer, but I do think it's quite interesting that not all mothers were aware, given the importance of antibiotics and emerging resistance, which of course is in a lot of conversations that we have in this evolutionary medicine community. So zooming into hand samples with these results, we found that these factors, maternal antibiotics and having been bathed prior to swabbing, were correlated with a higher abundance of Rhizobiaceae, which is a bacterial family that's often associated with nature, a microbe that's often found in soil, water, other parts of the natural environment. And then zooming into the cheek samples, we found that having that early hospital bath, so again, prior to me swabbing baby, was positively correlated to that same taxon, Rhizobiaceae, but is also associated with a lower abundance of staph, which we know is a large family that contains many different species, some of which are important commensals that are really key for immune system development. All right, so some of the results from the published paper suggest that we may be able to detect initial differences in the skin microbiome based on bath status. As an extension of some of that work, I've more recently been thinking through how best to measure differences in the skin microbiome at this time when babies are trying to pick up microbes from the environment for the first time. And so I've been thinking through which sorts of standard microbiome metrics actually matter in this context. So we can think about differences in diversity, right? Are there fewer taxa on newborns who were bathed prior to sample collection? We might also see differences in prevalence. Are there certain taxa that are just more or less common on newborns who had already received this bath? Now, I'm going to talk through some of these preliminary results, but I do want to say the really important caveat here is not just a relatively small sample size, but that I do not have paired samples from the same newborns. So I'm comparing different newborns, those who were just sort of accidentally, naturally unbathed before I got to the room to sample their skin, versus the group of newborns that had already received their bath before I swabbed them. With those caveats in mind, we can see that the skin microbiome of the bathed newborns does trend toward lower diversity and richness. So that's shown in these two plots. Though, of course, there is a pretty substantial spread or range of those data points in both of the groups. I also quickly looked at this idea of differential relative abundance. So here we're detecting a signal of differential abundances of bacterial genera in the newborn skin microbiome, again, based on bath status. So what you're seeing here is the taxa whose relative abundances were significantly enriched or depleted, that's either the dark or the light blue, on the skin of newborns who had already been bathed compared to those who were unbathed. Now, the plots you're seeing here are really from the same analysis, just the difference being where we put that P level threshold and what pops up as significant or not. Again, just highlighting it's an important metric to consider, especially with some of these studies with smaller sample size. So I then wondered more about the prevalence of different taxa, right? Operating under the idea that the newborn skin is trying to acquire new taxa in the first hours of life, I wanted to see if certain taxa were detected more often on the skin of newborns who had not yet been bathed, Or maybe certain taxa were more common on the skin of newborns who had already been bathed, maybe due to changes in those ecological niches on the skin following the bath. So first up, the newborn hands. In the heat map on the right, the darker blue cells are bacterial genera that were less prevalent on the skin of bathed newborns. So I'm highlighting here a few taxa with the strongest magnitude of differential prevalence. That's what you're going to see with the black arrows. In the same heat map, the darker red cells are showing us the bacterial genera that were more prevalent on the skin of the bathed newborns. We can also visualize the change in taxa prevalence between the two groups using this sort of a box plot, where you see sort of all these different lines crossing with each other. So this is showing us that certain taxa increase in prevalence between the groups, certain taxa decrease. So moving on to cheeks, again, blue in the heat map, showing us the bacterial genera less prevalent on the skin of bathed newborns. I do want to point out that for this lactobacillus that we detected, that's following the same pattern that we just saw on the hand samples. And again, the red is showing us bacterial genera more prevalent on the skin of newborns who had already been bathed. And then finally, our armpits. So again, blue in the heat map showing us bacterial genera less prevalent on the skin of our bathed newborns, and red showing those that were more prevalent on the skin of newborns who had been bathed. Again, on that list, Ralstonia is one of the taxa that showed the same pattern in these armpit samples and in hand samples. So a little bit of shared overlap in terms of patterns between these body sites. Now, one thing I want to do is bring these heat maps all together. I know this is a purposefully busy plot, but if you sort of zoom out a little bit, I do think there are some interesting patterns to detect here. So if we bring these three heat maps side by side, one of the things that we notice is that there are fewer taxa in the heat map of the armpit samples, right? That's the one on the right. We expect this because we already talked about how this body site has lower richness, has lower diversity, it has fewer taxa on it. The other thing to notice is that in that heat map of the armpit samples, we have more taxa that were less prevalent on the skin of bathed newborns, again shown in blue, compared to unbathed newborns. Now, something that I want to raise as an interesting point, and what I've seen in other analyses that I've done with this cohort of babies that I worked with during my dissertation, I tend to see this sort of pattern where the armpit often displays some different trend compared to the other skin sites where I collected samples from. So in this particular example, what I wonder is if the armpit actually has a harder time recovering from the potential disruption of the bath. In other words, even if all skin sites were bathed in the exact same way, you could speculate that the hands and the cheeks of newborns are more exposed to microbes in the hospital room and also more exposed to potential microbial transmission coming from other people who are coming into contact with baby after the bath. Armpits, on the other hand, may be more protected from some of those microbial transmission events, especially if baby is clothed or wrapped up in a blanket, which was the case for a lot of newborns in this study. So, to summarize what I've mentioned so far, we detected variation in the microbiome across skin sites within the first few hours of birth, potentially because of differential contact with different skin sites that could be independent of whether a baby had received a bath or not. We also found that certain modern hospital birth practices, like maternal antibiotics and the newborn bath itself, could be altering newborns' early microbial exposures. I also showed this emerging pattern of differential diversity, prevalence, and relative abundances across skin sites that may be shaped by different caregiving behaviors, including the bath. And so I think this raises an important question of if we should be thinking about the early bath as an additional driver of microbial mismatch, maybe something that works alongside everything that we've already learned about the disruption due to C-section birth. And if that's the case, much like we've seen in research with C-section babies, what could be strategies to mitigate some of this mismatch? Is that delaying the bath, or is there some intervention to re-inoculate babies after the bath? Again, like we've seen in the work on C-section. So I have a number of questions that are on my mind. I purposely left time so that we can talk about these today and any other questions that you may have. In terms of future research, I'm quite curious to test if that first bath is actually directly removing microbes from the skin. This, of course, would require a direct pre and post comparison from the same babies, right? So having paired samples before and after the bath. I'm also really curious what sorts of details of the bath might matter in this context, right? All I knew was whether a baby had been bathed or not. I didn't know anything about what they were being bathed in, who was performing the bath, what's the intensity or duration of washing the newborn's body. Is there variability if different nurses are performing that bath? I'm also curious how what I observed in this particular hospital might compare to contexts where the newborn bath looks different, right? The newborn bath may be delayed or just all around performed differently. This could be because of different practices in different hospitals, parents' preferences, but also cultural norms surrounding the initial washing of a newborn. One thing that's also on my mind, but I'm not quite sure how to incorporate this, and it wasn't a part of this work, is how does the bath intersect with either removal or retention of the vernix? That could be sort of two different things, and maybe parents have opinions about one or the other or both or neither, right? So that may be doing something to skin microbes in this context as well. And then, of course, there's this need to actually integrate immune or other health biomarkers so we can really understand some of these health impacts and understand if we may have a potential mismatch here. With all of these questions, I also want to present the question of, do these data actually matter for folks who work in these clinical contexts, right? Can we do this research and can we actually inform hospital bathing practices? Can we learn from what's been done with the research on C-section babies? But might it also be the case that some of this work on bathing practices is going to be more palatable for clinicians and for new parents, as opposed to, for example, a fecal microbiome transplant into a newborn's body? So I would love to talk about these questions and any others that you may have. I'll quickly just say thank you to the study families, nurses, administrative staff at the hospital I was working in Chicago. Thank you to my supervising physician, Dr. Emily Miller, my collaborators, co-authors, and the wonderful trainees who did a ton of lab work for this project. I also say thanks to all of you for listening and looking forward to the conversation that follows.
Thanks very much, Melissa. That was terrific. And I just want to just start things off by sharing how much I liked how many great questions you ended with. And I suspect that some of those questions are in mind from some folks online here. I'd like to start things off to invite the people who posted questions in the chat to present their question, unmute and present their question themselves, beginning with Nilusha. Are you still online?
Yeah, it's Nilusha. Great
Nilusha, thank
presentation.
you.
Yeah, great presentation, Melissa. That's really interesting. I wanted to just quickly understand if you have collected metadata or if you know whether these moms get to hold the babies right after they were born. And what do you think how would that play a role in changing those skin microbiome colonization trajectories? What are your thoughts about it?
Yes, great question. Unfortunately, I do not have those data. So just for a little more context, unlike some of the C-section studies where researchers were really there at delivery and sort of intervening or collecting samples immediately, I was basically walking into hospital rooms on the postpartum floor and just sort of saying, I was able to see some information on the electronic medical record. I know that you had a baby today, a few hours ago, and sort of walking into rooms and starting to recruit parents and newborns sort of at that time. So while I did catch a lot of babies in the first few hours of life, it wasn't early enough to know exactly what was happening sort of right after delivery, especially in terms of your question of, was mom or anyone else able to hold that baby immediately? When was skin to skin contact initiated? And again, if those things did happen, what would happen if baby then had a bath a few hours later, right? Again, I think, and this is, I think part of where your question was going, even if we have data on all these practices, what if we sort of have this unknown disturbance of the bath? Does that take away from some of these things that we generally believe to be really good newborn care practices? So it's a great question, definitely something to think about in future spin-off projects.
Great. Jillian, are you still online to ask your question?
Yes, I seem to have a little bit of an unstable connection. So sorry, I'm just going to keep my video off in case I get lost. But thank you, Melissa. That was really interesting. And I was just wondering, are there any comparative data on home births versus hospital births and how that environment might make a difference to the baby's developing microbiome?
Yeah, great question. I'm not familiar with much data in that space, but definitely something that needs to be explored, right? Again, I was working in one particular hospital context that did things a certain way. That may be similar to other hospital contexts. It may not be, but of course, it's this other population of babies that are being born in a very different context, and one that I would assume is maybe richer in microbes than a hospital that we try to sterilize, that we try to clean, where we try to reduce the bacterial biomass in those spaces. So I would suspect that it's a different pattern of initial colonization in a home birth. I think an additional interesting question is what about a water birth, right? Similar questions of what are sort of those initial microbes, where are they coming from, but then what's happening soon after that sort of first inoculation? Is it taking away from that rich initial exposure that's happening at home? Or maybe in the context of home births, baths are also more likely to be delayed or at least performed differently than they would be in a hospital setting. It's a great question.
Thank you.
Thanks.
Excellent. I see there's a question from Wenda. Are you
I,
still
I
online?
am. I am.
Great.
Thank you.
Thank
Yes. Thanks, Melissa. I thought this was really exciting stuff. One of the first things I think about when I think about bathing a baby is removing the vernix. And in fact, I think the World Health Organization a few years ago recommended against immediate bathing for the very purpose of the protective value of vernix. And you did mention it as a question to consider in the future. But what do we know about whether vernix actually helps to sort of prevent the absorption of microbes because it is preventing a lot of other stuff, or whether it actually works in favor of getting those microbiota at that
It's
time?
a great question. Again, I'm going to say I don't think we know very much, which is clearly a common theme of this Q&A, but I think an important theme. I have thought a lot about this, and it appeared in the EMPH paper that was referenced. Very limited data, but of course something that I thought of immediately as well. Again, might parents have a preference on vernix removal that is also related to a full-blown bath? Are those preferences correlated or not? Are preferences actually really asked and honored sort of for both of those things, at least in a large hospital setting? But also more to your question of sort of what's the underlying ecology that's happening with the vernix. I don't think we really know from a microbial perspective. What I suspect matters is what vernix retention would do in terms of some of the environmental factors of the skin that we know bacteria are sensitive to. So, moisture, skin hydration, pH, some of these factors that we know sort of in environmental samples, like in soil, bacteria are really sensitive to changes in those parameters. So, I think that's where I'd be curious to sort of dig into the idea of the vernix shaping the skin microbiome really by tinkering with some of those parameters and then potentially sort of, selecting for some of these bacteria that are better adapted to a more moist environment, right? Or skin of a certain pH. So that's, I think, the logical entry point where I would like to go with that.
If I could sort of tag on to that, could
Yeah.
that possibly be modeled in the laboratory?
Hmm.
I mean,
Yes.
these are really good questions, but
Yeah.
these are also complicated studies. I mean, I don't need to tell you that, and you're always going to be wrestling with problems of power. And I'm wondering if some of these factors could be modeled.
Yeah, that'd be fantastic. Yeah, I mean, can one culture vernix and throw microbes into it and see what happens?
I mean, this is literally what we're, yeah, or an extraction or something. Yeah.
Right, right. I would love to do that. Absolutely.
Um, Handing over, there's a provocative question from Antonio, if you're still online.
Hey, Melissa. I mean, I'm sorry. Hey,
Hi,
Dr.
Tony.
Melissa.
Hi, Dr. Baines.
I It's so cool to see you. So cool to see you. So cool to see you doing your thing. Congratulations. Congratulations. So
Thank
yeah, great presentation. So, as you know, for many years I have an interest in health disparities. And in terms of births, African American women, die more than other women in childbirth, for a variety of reasons. So I'm just curious if you think the microbiome could be playing a role in some aspect of that. Knowing that the baby and the mom is very highly connected during that time. Um, so yeah, I'm just curious, just curious, especially when you talk about C-sections and vaginal births and,
Yeah.
yeah. So I'm just curious to hear your thoughts. And again,
Yeah.
congratulations again on your success.
Thanks, Tony. Yeah, that's a great question. I mean, I think, obviously to answer any of these questions about health outcomes and importantly health disparities, there has to be this link between the microbes that we're getting really good at sequencing and measuring and detecting and understanding, and real life health outcomes for babies or mom as well, in the case of your question. I think that's where, again, I alluded to this. I think, where a lot of this research, even beyond just this little world of the newborn microbiome, but it's where a lot of this microbiome work has to go. Again, we've gotten quite good at figuring out the microbes that are there, but I think the really key step for understanding what this means for human health and health disparities is what are these actual biochemical pathways through which these different microbes are really interacting with host physiology. And in the context of childbirth, how's that interacting with all the various stressors, right, in the body, everything that's going on, hormonal changes, right, all these radical biological shifts. How are the microbes sort of sitting along those pathways is where a lot of this has to go. I will also say, is a bit tangential to your question, but I did think it was really interesting. So, I mentioned I was recruiting babies from a particular hospital in Chicago, and I was really trying to exclude C-section babies, right? For all the reasons we talked about, it was going to be a lot of noise in this data set. I will tell you, I had a really, really, really hard time recruiting families of racial diversity, socioeconomic diversity, diversity in mom's age, diversity in which zip code in the Chicagoland area families lived. I had a really hard time capturing a broad range of those sorts of demographics when I, for good reason, excluded C-section babies. I think you can probably fill in the gaps of what I'm alluding to here. But it was really quite difficult, and something that didn't really surprise me, but when it's happening, it's right there in your face looking through these medical records, and it's pretty alarming to see, especially at a really great teaching and research hospital in an otherwise very diverse city. And so that was a real challenge for this project, and it's frankly logistically part of why the sample size for the hospital babies is lower than what I wanted it to be, because I had to leave the hospital, and I had to go to different clinics in different parts of the city and surrounding suburbs that were really serving a more diverse patient population. But logistically and sort of timeline for me to wrap up my dissertation research at the time, I was then finding those infants much later on. So these were infants who might have been multiple months old, and I was meeting them at their primary care or pediatrician's office. So again, trying to exclude C-section babies, but dealing with what other sorts of variables does that also automatically exclude? And so having to be creative about what you can really intentionally exclude, or if at some point you sort of have to bring that variable in and control for it so that you know you're capturing enough variation in other parameters. So I know not directly related to your question, but I do think it's an interesting point and something I continue to think about as I think about building a new cohort of newborns. What does that need to look like, and how do I find these babies, and making sure that it's representative of the actual population, in the location where I'm working.
Um, unsurprising for maybe I hopefully unsurprising to you, you've stimulated quite a lot of interest in the chat. So I'm going to take the liberty of summarizing the next two.
Okay.
So Patty Medina wrote, it'd be interesting to look at the microbial environment in twins and multiple births. Did you have any of that in any of your studies?
No, it's a great question.
I figured not, but a good one.
Yep.
Add to your nice list at the end.
Yeah.
The next one is from Patrick Lothrop, who couldn't unmute today. Some C-section babies wind up in the NICU postpartum. Are there data on time of bathing between those who do not end up in the NICU and those who do not?
Another great question. I'm not sure if there are any clinicians on the call who do know. If they have a general answer, please let me know. I know for me in this particular study, I was excluding babies that had been in the NICU. I was excluding anything that was flagged on the medical record for mom and baby having some like larger flaggable medical condition, especially because this was also in spring of 2021. So if anyone had a positive COVID test, I wasn't going into that room for multiple reasons. Right. But a lot of the health conditions that might be flagged, making mom or baby high risk, are probably also conditions that would affect the microbiome in some way. So I was also excluding those babies. So again, no NICU babies for me in particular. I am curious though what that means for the bath in general. If anyone knows the answer to that, please let me know.
I'd like to call on Michelle Blythe if you're still online. Your question hit a nerve. It appears on the chat.
I am. Yeah, so I was just sitting here thinking about this, and I'm like, well, mom's microbiomes, aka ours living in this current quote unquote modern society, we already know is kind of different from the more traditional societies that we study and all of that. So I guess my question that kind of posed is, are these health outcomes and everything determined by what we're giving our infants? If our microbiomes are already weird, are we just giving weird microbiomes to our infants? I don't know.
Yes, we probably are, I think is the short answer. Yes, right. And we can think about sort of the uh, influence of drastic environmental changes on the microbiome that we can see across different generations, including microbes that may be directly passed on between generations, right? Including from mom to baby. I know some folks have been thinking about this in the milk microbiome space, which is also where some of my work lies. I mentioned B. infantis as this really key species that is transmitted through milk and is really good at colonizing baby's gut, setting up shop, training the immune system. Because it's so important, we just sort of figured that we should see this species in pretty high abundance in all milk samples collected everywhere and in all babies' guts collected everywhere. And that's totally not the case at all. And so this has been a really interesting question, especially for me, because a lot of the folks I work with in the milk microbiome space are not trained in human evolution or anthropology. And so having these conversations of, if something is so important for baby, why are we not seeing it everywhere? Why would there be variation? It really brings up questions of even some of these microbes that we consider to be so human associated and so critical. They're still coming from somewhere, right? They're coming from somewhere in the environment. And so I think probably most people on this call would agree that to think about even these really important human microbes, we have to consider that environment that mom, baby, anyone else is living in, right? And how that environment may or may not have changed over the generations for that particular mother-infant dyad in question. But to Michelle's point, yes, I mean, it is very possible that some of these urban industrialized moms already have weird microbiomes, and that's what's being passed on. And that's a whole other issue outside of what kind of bath we're giving baby or when that's occurring. And I think that's something that needs to receive more attention. Again, the acronym of weird societies. Maybe we also all just have weird microbiomes, and that's more important than anything else here. Maybe.
Um, Thanks, Melissa. I'm gonna, in the interest of time, let Karen Rosenberg pose the last question.
Okay.
You
Thanks.
up for it, Karen? Yep.
Yep, thanks so much. That was really wonderful, Melissa. Thank you very much. So this is kind of a classic anthropological topic, right? Washing babies. After all, Margaret Mead made a bunch of ethnographic films, I think in New Guinea, about how people watch, admittedly, older babies than infants. But I was wondering if you'd looked at the sort of ethnographic literature to see what cross-cultural variation there is in people's ideas about what's appropriate to do with little babies.
Yes, that's a great question. It was not a part of this particular project, but yes, has been something on my mind, something I've been thinking and reading about, and just sort of talking to people anecdotally as much as I can. Because again, I think there's sort of different scales of variation where we can think about this. You could have variation in these early bathing practices, potentially just within the same hospital, right? Depending on what mom is asked and what sort of preference she might have. You could have variation just on the same floor, right? Probably more likely that you're going to have variation sort of between hospitals, sort of between these birth environments, certainly within the US. And again, some of that may just be sort of standard practices in that hospital or in that environment where baby was born. But I think probably more interesting to a lot of us with an anthropology background is maybe less, what's sort of happening in the background because that's the hospital practice, but this interest in what are some of the longer standing cultural practices that are more intentional in terms of how baby is bathed, right? So maybe not just a question of a delayed bath or not, but exactly when baby is bathed, depending on the cultural context, is it just water? Is that water infused with other things, right? Herbs, things like that. I think a lot of questions to ask in this space. Of course, the difficulty would be pairing ethnographic data or things you can find in the literature that's, was probably those data were collected at a time when no one was collecting microbiome samples simultaneously. So that, of course, is a challenge to, understand how that might be shaping some of the data we're collecting. But in my grand vision of sort of a global cross-cultural anthropological study of newborn bathing, I think, asking those questions and really actually measuring properties of the water that's being used, what's in it, the timing of the bath, who's performing that bath, is it delayed? I think all of these are really important considerations.
Outstanding. I just want to maybe end by offering to you the value of this network if you weren't aware of it already. I know you are, but
I
that
am.
many of us are at major medical centers and I've been sitting here thinking all this time what's going on down the road at McGee Women's, which, basically where most kids in Pittsburgh are born.
Hmm.
And then obviously what also is going on over at the Children's Hospital, which it would be more intensive, more sort of interventionist care. And I mean, I
Yeah.
can get that data, and I know many of us here represent our own institutions and probably can get that data. So always consider it. I'll hand it back
Yes.
over to Charlie. Thanks again. This has been really great.
Thanks, Vaughn.
Yeah, I'll close things up by just saying thank you, Melissa. That was really fantastic and generated so much great discussion. I learned a lot from your talk and in the discussion afterwards. So thank you. Vaughn, thank you very much for nominating Melissa and for recognizing her paper in EMPH. And I hope everyone will check out the journal and consider it for your papers. And thank you all for tuning in. It's great to see so many of you here today, and we have another one next week. So stay tuned for that. The announcement will go out soon. Thanks, everybody.
Thanks, everyone.